Okay, so welcome to this video on the pathogenesis of Black Death. I'm hoping to do a whole series of videos on pathology, and we're going to start with this bacterium. It's one of the more dramatic diseases in pathology. It is really, it's one of my favourites. Obviously, it's absolutely ghastly if you get it, but as far as learning about it is concerned, it is quite dramatic. In the Western world, you know, we're worried about atherosclerosis, and the diseases that terrify people are HIV and cancer. HIV takes 10 years to kill most people. This Black Death will kill you in 10 days if you get it. So, without further ado, let us begin. So, the bad guy in this story is a rat. The rats are one of the ways that humans became infected with this bacterium. Firstly, the bacterium that actually causes HIV is called, I'm going to write this down, it's a bit far away, I need to alter this. Okay, so there we go. Well, the bacterium that actually causes HIV is called Yersinia pestis. Yersinia pestis, and it's one that generally no one has ever heard of before they go to medical school. So Yersinia pestis is the bacterium that causes Black Death, and so the story begins with a rat. So the rat is infected with Yersinia pestis. So we have a rat. Am I going to embarrass myself by trying to draw a rat? Yes, I am. How now to do? Ooh, there we go. That's a rat. It's got some legs. The legs have ruined it, but never mind. There we have our rat. So our rat is infected with this bacterium, and this bacterium, just so that you can visualise it, is a sort of rod shape, and it's gram-negative. So why have I drawn it blue? Damn it. It should be red. If you gram stain it, it appears red, so that means that it doesn't have a very thick cell wall. So it loses the, oh god, what's the crystal violet stain. So in the gram staining process, you firstly stain it with the crystal violet stain, which stains it blue, and it stains anything that has a cell wall blue. But you then add iodine, which fixes the stain into the cell walls, but the stain only really remains in the cell wall if the cell wall's really thick. So gram-positive bacterium that appear blue when you gram stain them, they have very thick cell walls, which is the reason the stain remains there, so that you see them as blue. Whereas gram-negative bacterium, when you wash it away, you then wash the bacterium in the gram staining process with, I think generally acetone, which is a powerful solvent. So that washes away the stain from the gram-negative bacterium, which have a very thin cell wall, but not the gram-positive bacterium that have a very thick cell wall. You then add a counter stain, which is safranin, which is a red stain, and this stains all the bacterium with the cell wall again, but the blue stain is so powerful that it overpowers the red stain, and you don't see it in the bacterium that's still saying blue. So only the bacterium that lost the blue stain appear red. So those are the gram-negative bacterium. So this is a gram-negative bacterium, so it's going to appear red. It's going to appear red. Okay, so now this rat, the story gets worse with this poor rat. The rat might have a flea on it. How do I draw a flea? Fleas kind of look like a blob with big legs and a sort of head, I think, there. So the flea sort of drinks the rat's blood, or whatever it does, and it becomes infected with Yersinia pestis too. So the Yersinia pestis is in the rat and it's in the flea. And I don't know generally what happens, but I would guess that it's the flea generally that actually spreads it to the human rather than the rat. But one of these two things bites the human. So they come along, and here's our human over here. And they're going to bite the human. So they're going to bite the human, and this bacterium is going to go into the human's skin. And then what happens? Now, if I took some bacterium and put it into your skin, what would usually happen? It would get obliterated by the macrophages that are resident within your skin. So why does this bacterium not get obliterated by the macrophages in your skin? Well, it has a number of clever tricks up its sleeve. So if we have a macrophage here, which is a big blob of a cell, so there's our macrophage, and here, well, our bacterium will be tiny compared to the macrophage, but we'll draw it that size there. Well, basically, firstly, our bacterium has a capsule around it, which is antiphagocytic, so it means that the macrophage is going to struggle to to phagocytose this bacterium because it's got an antiphagocytic capsule around it. An antiphagocytic capsule. So that's the first reason that the bacterium can't struggle to phagocytose this bacterium. And secondly, this is rather cool, this bacterium makes a little needle, basically, so a tube made out of protein, and it sticks it into the macrophage, so it sticks it through the cell membrane, the phospholipid bilayer, and then it sends little proteins through the tube into the macrophage. And these proteins, I don't know how they work, I don't care how they work, but they stop this macrophage phagocytosing this Yersinia pestis bacterium. And I don't know, maybe they interfere with the actin, because to phagocytose, you obviously have to change your cytoskeletal structure so that you sort of wrap around the bacterium, and if you can't, I know that tuberculosis or is it TB? Ooh, I think it could be Shigella, actually. So I know that a target for preventing phagocytosis is to prevent the actin remodeling that you need in order to phagocytose this bacterium. I think it might be I don't know, actually. Or it could be Salmonella. Some bacterium do target the actin cytoskeletal remodeling to prevent the macrophage from phagocytosing them. So Yersinia pestis has pulled this trick on us, so it's prevented our immune system destroying this upon entry. And now what's going to happen is that the Yersinia pestis is going to divide in your skin. So the Yersinia pestis here and here divides in the skin, and it goes into the regional lymphatic vessels, which then drain it to the regional lymph node. So Yersinia pestis comes to a lymph node, so we'll now draw a lymph node. So lymph nodes kind of look like, this is going to be awful. So here's an afferent vessel, here's another afferent vessel, here's another afferent vessel. So it comes in like this, and then there's one efferent vessel that comes out, so this is the efferent lymphatic vessel. There will be far more afferent lymphatic vessels than I've shown here. Efferent vessel, and those other three vessels are all afferent vessels. Afferent vessels. Afferent vessels. So around the outside there is like this sort of, how am I going to draw this? There's a sort of like sinus where all the lymph sort of comes in, and it sits in this sinus. And then underneath that there is a layer called the cortex, and in the cortex there are B cell follicles, which we've drawn here, and around the B cell follicles there are T cells, and then it sort of goes into a medulla where you've got these medullary cords coming in like this. And then it comes into a central sinus which drains into the, I don't know what kind of happens here. I think it must sort of go like that. And it sort of, all the lymph basically has to percolate through all these lymphoid tissue and to get to this medullary sinus, I think this must be called. Medullary sinus. And then the lymph, once it's in there, goes out of the afferent vessel. So in this cortex you have the B cell follicles, so this is where all the B cells live, and surrounding that you have all the T cells living here. And the lymph comes in, percolates through this marginal sinus, as this is called, and then goes into this this lymphoid tissue. So what's going to happen is that the Yersinia pestis is going to come in, and it's going to go and it's going to percolate through this lymphoid tissue, and it's going to get stuck there. And it's going to cause a major inflammatory reaction there. It causes something called hemorrhagic inflammation of the lymph nodes. It basically makes you bleed into your lymph nodes, and your lymph nodes swell to an enormous size, and they're then called buboes. Buboes. Or a bubo is the singular. And you are then said to have bubonic plague. Bubonic plague. So you get these huge, great, swollen black lymph nodes called buboes. Plague. And you are then said to have bubonic plague. And you can only catch bubonic plague from a rat or a flea. you cannot catch it from a human. If you catch Yersinia pestis from a human, you get pneumonic plague. You don't get bubonic plague. As we'll see, bubonic plague becomes pneumonic plague, but if you catch Yersinia pestis from a human, you will never get bubonic plague. You will just go straight to pneumonic plague. So this is a step that only those who are privileged enough to catch this from a rat will go through. Okay, so that's that. And by the way, the cause of this huge inflammation is that this bacterium is gram-negative and therefore has a lipopolysaccharide in its cell membrane, in its outer cell membrane specifically. Gram-negative bacterium have two cell membranes and a space in between called the periplasmic space. So Yersinia pestis releases this horrible molecule called lipopolysaccharide. It doesn't release it deliberately, it's just in its membrane, and vestiges of it go off and go off into the ether, and it's that LPS that causes this horrendous immune response. So lipopolysaccharide is that. It's also called endotoxin. So the difference between an endotoxin and an exotoxin is that a bacterium produces an exotoxin deliberately to attack the host. So an example is like pertussis toxin, tetanus toxin, botulinum toxin, cholera toxin, Shiga toxin. These are all foul exotoxins. Staphylococcus aureus produces a toxic shock syndrome toxin, and I think Streptococcus pyogenes produces a toxic shock syndrome-like toxin, which are superantigens and cause a huge immune response. But this is not an exotoxin. This is something that the bacterium makes for a totally different reason, because it wants it in its outer cell membrane, and it just happens to cause a horrendous immune response, which causes most of the pathology associated with the bacterial infection. Okay, so now from the lymph node, the bacterium is going to go into the blood. So we've had this huge hemorrhagic inflammation, so the blood vessels are breaking apart, basically. So the bacterium can go basically into the blood easily, because the blood vessels are no longer sealed, so it can just sort of go into the blood vessel, and then it's in the blood, and it is going to drain. It's going to drain into, say we got bitten on the arm, then it will drain into the subclavian vein. So shall we do a bit of revision of anatomy? Yeah, it will drain into the subclavian vein. That will then join with the internal jugular vein, and they'll make the brachiocephalic vein, which will join with the other brachiocephalic vein to make the superior vena cava, which will go into the right atrium, which will then go into the right ventricle, and the right ventricle will pump the blood into the pulmonary trunk, which will split into the two pulmonary arteries. So the Yersinia pestis is coming in this way. It's going in through the right ventricle, it's going into the right ventricle, it's going up into the pulmonary trunk, up into the pulmonary artery, and then it's going to go into pulmonary capillaries. And then if we have an alveolus drawn here, so these are the alveolar septa, so this is histological level. These are the alveolar septa made up of pneumocytes. These look like type 1 pneumocytes. And we'll have little blood vessels running in the middle. And basically what's going to happen is that the Yersinia pestis is going to leave the blood vessel and go into the alveolus. So the Yersinia pestis is now in the alveolus, and it basically makes itself at home, colonizes the lung, and you now have a pneumonia infection. Pneumonia means that you have a bacterial colonization of the alveoli. So you have pneumonic plague now. Pneumonic plague. Excuse me. Sorry about that. Pneumonic plague. Quite relevant that I just sneezed, therefore. So pneumonic plague is the one that, once you've got pneumonic plague, you can actually pass it on to other humans. So you're going to cough up sputum, and your lungs are going to get hugely inflamed. So you're going to get, basically, now once the bacteria has colonized, it will start destroying tissue of the lung. You'll get necrosis. That will release ATP. ATP will activate pattern recognition receptors on the macrophages, as well as LPS will activate pattern recognition receptors. I think it's specifically toll-like receptor 4. those will activate the macrophages. The macrophages will release tumour necrosis factor alpha, which will go and act on the endothelium of the pulmonary capillaries. The endothelium will contract. You're making huge spaces in the capillary wall. Exudate will pour out of the blood. You'll get fibrinogen everywhere. Fibrinogen will convert into fibrin because the coagulation cascade will be activated, because factor 7 will meet factor 3, and all of that will happen, and you'll get coagulation. Fibrinogen will turn into fibrin monomers, which will assemble into fibrin chains, and you'll get basically an acute inflammation. In addition, tumour necrosis factor will make the endothelium express a huge number of sticky molecules that attach polymorphonuclear leukocytes, neutrophils is the new name for them. They'll attach to the surface. They'll move by diapedesis across the endothelium into the alveoli. You'll get a huge inflammatory exudate of neutrophils, fibrin everywhere, and you'll get lot of other things as well, which I'm not going to go into because it's painful. But you will get, but the end result of this, of course, is that you will have reduced gas exchange. So if it's bad enough, you will obviously get cardiogenic shock, because you're not going to get enough oxygen into your blood to meet the demands of your body, and you'll, become breathlessness. You'll get breathless. You'll get cyanosis, and you'll get fatigue, and you'll eventually die. And also obviously this bacterium's destroying your lung as well is another problem. In addition, now it's at this stage you can actually pass it on to another human being. You're going to end up coughing up sputum. You're going to sneeze. You're going to be coughing up loads of this bacterium, and if other people breathe that into their lungs, then the bacterium can colonize their lungs, and they will get pneumonic plague. But they will not get pneumonic plague in the same way you got pneumonic plague. You got it through bubonic plague by getting it from a rat. They will go straight to pneumonic plague. They will not get bubonic plague. Which is, I don't know whether that's some relief for them, but this is what kills you. It's not the damage to the lymph nodes that kills you, it's pneumonic plague, pneumonia. There are other ways this bacterium kills you, and we'll go over them now. So, from the, we've gone into the right ventricle. It'll go through the pulmonary artery, through the pulmonary capillaries. Not all of the bacterium will leave the blood at the pulmonary capillaries. So some will go back into the pulmonary vein, into the, so through the 4 pulmonary veins. There are 4 pulmonary veins. So they'll come back through into the pulmonary veins, and they'll go into the left ventricle, shown here. And the left atrium, shown here, and then into the left ventricle. It's not very heart-like. It's usually better than that, my drawing of the heart, but this is wrong. It should be like, yeah, I'm going to correct that. I'm going to draw it there. Ignore that. That's slightly, this is a bit too extreme as well. But here's the left ventricle, basically, and the left ventricle is going to give off the aorta, which comes off up here. Because the left ventricle is going to go behind the right ventricle, so the aorta comes off to the right of the pulmonary trunk, and it's going to go round into the aortic arch, and then it's going to throw off the brachiocephalic artery, the left common carotid, and the left subclavian artery, and then go down into the thoracic aorta. So we're going to start with this one. It will go, the brachiocephalic splits into the right common carotid and the right subclavian, and it's going to go up these two here to the brain, basically. Bad news. So it's going to go into the brain, it's going to go into capillaries in the subarachnoid space. So just a bit of a reminder here. Here's the pia mater, this is the bottom layer. Then we've got the arachnoid mater, and above that we've got the 2 layers of the dura mater, potentially with a sinus in between them. At some points of the brain there'll be a sinus in between them. So for instance, the superior sagittal sinus is a sinus of this form. But obviously sometimes these 2 layers will be confluent and there'll just be 1 layer. So where are we? So this space here is full of CSF, and it has blood vessels in, and the Yersinia pestis is basically going to come out of these blood vessels and go into the subarachnoid space. And it's going to colonize the subarachnoid space, and it's going to cause meningitis. Meningitis, and that's dangerous, really dangerous. because what's going to happen now, it's going to cause necrosis of some of these cells of the pia mater and arachnoid mater, and then what's going to follow that, you're going to get inflammation. And inflammation in the subarachnoid space is not a good thing, because this is going to swell, and what's it going to press down on? What's sitting here? The brain, which I'm just about to attempt to draw. Goodness, what happens at the bottom? Just that's close enough. Oh dear, that's close enough. And so it's going to push down on the brain, and it's going to cause brain damage. If not, meningitis does. If you survive meningitis, it usually does leave severe neurological impairments. But usually it's fatal. So if pneumonia doesn't kill you, meningitis is going to kill you. And if meningitis doesn't kill you, we're now going to go on to another way this bacterium can kill you. So actually, before we go on to the the final way it can kill you, which is multi-organ failure, we'll discuss why it's called Black Death. So I will get another piece of paper. So this is quite a nice little story. So this is why it's called Black Death. Here we go, like that, I think. What colour do I want? I don't know if black's showing up particularly well. I might go back to the big blue pen. Okay, so we need our diagram of the circulation back again. Okay, so what if it goes into the... So instead of going up to the brain, it can also go into this artery, so it's disseminated all around the body. So this is the subclavian artery, the left subclavian artery, which is going to carry blood to the arm. So let's just have Yersinia pestis coming to the arm, and let's say it goes to the skin. So it came in through the skin and it's gone back to the skin. So it's going to go to skin all over your body, and it's going to colonize skin in your body. So what is this? This can be the hypodermal layer. Then we've got the dermis here and the epidermis here, and these are the epidermal pegs, dermal papillae, or is it the other way around? No, it's epidermal pegs and dermal papillae. So this is the epidermis. Epidermis. This is the dermis here, and this is the hypodermis at the bottom, which is just a layer of fat, basically. Hypodermis. So the bacterium is going to come in and it's going to colonize the dermis majorly, because there are lots of blood vessels in the dermis. So it's going to come in, and here's our bacterium, a bit out of proportion. And it's going to divide here, and it's going to cause, basically, it's going to cause the death of all the cells here. So you're going to get huge, great patches of necrotic skin, which means you're going to get huge, great patches of black skin on you. And that's why it's called Black Death, because it literally looks as though you are gradually dying, as though bits of you are gradually dying. You get these black spots all over you of necrotic skin, and obviously Google this, you can see the horrible pictures of this happening. And that's what's happening, that this bacterium is colonizing the skin, it's causing death of all the skin cells, and you are getting these huge black patches of necrosis, and that's why it's called Black Death, because it's of these black patches. And you're gradually turning to black and dying. So it's, yeah, that's why it's called Black Death. But obviously, the problem with necrotic skin is that the skin is your boundary against all other sorts of infections. So this is going to mean that other infections can go in. So if your situation wasn't bad enough already, you're going to get other horrendous infections. So it's just getting worse and worse. Okay, so another way Yersinia pestis can kill you, if we go back to the circulation diagram, is it can go down here, into the thoracic aorta. And at the level of T4, sorry, not T4, T12, what comes off? So if behind we have the vertebral body of T12, so this is T12, and just to emphasize it, we'll have the ribs, 12 ribs coming off here, 12 ribs. And here's the thoracic aorta. aorta, and now I'm testing your anatomy. What comes off at T12 at the level of the aorta? It's the celiac trunk. So here's the celiac trunk, and what comes off the celiac trunk first? The left gastric artery, then followed by, what comes off next? The splenic artery goes off down here, and then it becomes the common hepatic artery, and it's the common hepatic artery then gives off up here. It gives off the, oh, can you see this? It gives off, what's this? This is the what is this? This was the left gastric artery, so this is the right gastric artery. And I'm going to have to squash what's left in. And these two arteries that are coming off now and are the, what's this one? This one is the gastroduodenal artery. And this final one is the supraduodenal artery, I believe. Supraduodenal artery. And I don't know why I'm going through this level of anatomy for this. I've gone too quick. This bit that's left here is now the proper hepatic artery. I don't know why they called that the hepatic artery when all of this is still left to come off. This should have been the common hepatic artery. Okay, and then shall I go into this even more detail? The common hepatic artery comes up, bifurcates into two halves, and then each half further bifurcates. And this one is the left hepatic artery, this one is the middle hepatic artery, this one also bifurcates. This is the right hepatic artery, and this is the cystic artery supplying the gallbladder. Okay, so Yersinia pestis is going to come up here, and it's going to go into the liver. So the structure of the liver, shall we go? I don't know why I'm doing this, it's just pathology, but the way that pathology becomes interesting is if you use it as you have to make all of the anatomy and physiology and histology relevant to the actual pathology, otherwise you just forget the anatomy and physiology and histology. So here's a bit of anatomy, and now we're going to do a bit of histology. So the structure of the liver is that it has hepatic lobules, which are in like a hexagon shape to make it simple. And at each of these corners of the hexagon you have what's known as a portal triad, and at the centre of the hepatic lobule you'll have a central vein. So the portal triad consists of a branch of the hepatic portal vein, so you have one at each pole. And of course, you then have another, sitting here you'll have another hepatic lobule, and sitting here it all tessellates beautifully. So there'll be another hepatic lobule there, and etc, etc. So here are all the branches of the hepatic portal vein. You'll also have a branch of the hepatic artery running alongside, and then finally, what's the final one? A branch of the bile duct, which is the smallest. So basically blood from both the hepatic artery and the hepatic portal vein is going to percolate through the hepatic lobules to this central vein, and bile from the so in this hepatic lobule there are loads and loads of hepatic cells, hepatocytes, and they obviously are filtering all the blood, so they're removing bad things from the blood, but they're also producing bile, and the bile goes the opposite way to go to the bile ducts. Okay, so Yersinia pestis is going to come in through the hepatic artery, it's going to percolate through here, and basically it's just going to go into your liver tissue. It's going to start dividing, it's going to cause necrosis of your hepatocytes, it's going to then lead to liver failure. And it's going to do a similar thing to your kidneys and to other organs. So you're going to get multi-organ failure. And that's the final way that this horrible bacterium will kill people. So it is a really, really horrendous disease. I hope you've enjoyed the video, I hope you've learnt something, and keep loving pathology. Bye!